Hi, I'm Shanna Henk, and I'm going to be telling you about single-cell sequencing. Single-cell sequencing is exactly what it sounds like. It's sequencing the nucleic acids from individual cells. It can be used to compare genetic differences in DNA across cells, for example, changes in tumors relative to the surrounding cells. Examining the function of cells in their microenvironmental context by studying gene expression, or RNA, studying the differentiation of cells during development, or studying the DNA methylation and epigenetics. Single-cell sequencing is a next-gen sequencing technique with a setup that's very similar to digital droplet PCR. Instead of separating DNA fragments into droplets or tiny wells, like in digital droplet PCR, it separates cells. It is also similar in some ways to flow cytometry, which I'll talk more about later. There are different types of equipment for doing single-cell sequencing, but to explain it, I'm going to focus on mRNA dropseq, which uses droplets like in digital droplet PCR. Flow cells generate droplets within oil that contain a particle or a tiny bead, a cell, and the components needed for the reactions. For example, ddNTPs, buffer, reverse transcriptase, etc. Each particle is coated with a different genetically bar-coded oligo-DT. RNAs are captured on the particles. Within the droplets, reverse transcription reactions generate cDNAs with barcodes on the end that cause them to be stuck to the beads. They are known as stamps. Template switching at the reverse transcription stage generates multiple Cs on the end so that you combine primers to each end of your fragment. The cDNAs are amplified using PCR to generate a sequencing library. Finally, you sequence the fragments, which are no longer attached to the particles, and analyze the results. You can process many cells in parallel in a single tube so that you can sequence thousands of cells at the same time. Reads from individual cells are identified by their cell-specific barcodes. As I showed in the previous slide, dropseq allows many reactions to occur separately but in parallel by incorporating droplets as reaction chambers. Another way to perform single-cell sequencing is using microwells. Each chip can have more than 1 million wells. Particles and cells are washed over the chip so that the wells act as the reaction containers instead of droplets. The blue image shows a chip with black wells, blue particles, and red cells. After sequencing, you have hundreds of millions of reads. How do you make sense of it? The DNA has a cell-specific barcode that it picked up from the particle it was attached to, so reads can be sorted by cell. There is also a unique molecular identifier, or UMI, which is another short barcode that tags each fragment prior to PCR. After PCR, you will have multiple copies of that fragment, all with the same cell barcode and UMI. All of the sequencing reads from a single cell associated with a single UMI are pooled. This accounts for bias induced by exponential PCR amplification. Once reads are sorted, you can compare levels of gene expression across cells, like what you see in the table on the far right. The controls used in single-cell sequencing are very similar to those used for other types of next-gen sequencing. You want to have a no template control to check for contamination of your reagents. If you're starting with RNA, you also want to have a no reverse transcriptase control. The unique molecular identifiers control for bias in PCR since each run from a particular starting fragment has the same UMI. Thus, you can tell how many copies of a gene you had initially instead of this being changed by the vagaries of PCR. There are also quality control measures that are used on the sequence. For example, Illumina's sequence analysis viewer tools. And then finally, you can compare sequence to reference sequences and eliminate any sequences that are the result of contamination. The process has some limitations. One of the biggest has to do with sorting particles and cells into compartments. Only a small fraction of the droplets or microwells contain a single cell and bead. Most are empty or have only one or the other. A tiny fraction are multiplets, so they would have 2 cells and 1 bead, or 2 beads and 1 cell, and there's no simple way to detect or eliminate them. You can use multiplexing, but I don't have time to talk about that here. If you increase the concentration of cells and beads, you can increase the fraction that have a bead and a cell, but it also greatly increases the chance that you'll have more than one bead or more than one cell in a droplet. Another limitation is that it's more expensive and lower throughput than flow and mass cytometry, but it provides information about many more genes. The advantages are that you can process many thousands of cells in parallel at the same time. It also has the ability to compare expression of many genes to one another and tease apart the functioning of different groups of cells. So to show you some examples of its use from the literature, in the first case, single-cell sequencing was used to detect a mutation in a tumor cell. They were able to detect a switch from a C to a T, which is highlighted in the red rectangular section of the upper figure. In the second example, the authors were looking at different cell types within the retina of the eye. The left A panel is a schematic representation of the major cell classes of the retina, with different colors for the different cell types. In the B panel on the right, each dot is one of 44,808 single-cell expression profiles, and the clusters, which are colored by cell class, as shown in the A panel, are cells that are functioning similarly. In other words, they have similar transcription profiles. The plot shows a two-dimensional representation of gene expression among all the cells. They've reduced 32 statistically significant principal components into 2 dimensions with a dimensionality reduction technique. You can study expression of up to a few dozen genes at a time with flow cytometry and come up with something similar to the B panel on your right. However, with single-cell sequencing, you can look at differences among as many genes as you want at a time. In this final example, single-cell transcriptomes were collected from the malaria parasite, Plasmodium falciparum, at different stages of asexual development and sexual development, or gametocytes, within red blood cells. The A panel shows the life cycle of the parasite. In the B panel, on the top, you can see the distribution of unique mRNAs per cell and the total number of expressed genes per cell on the bottom half. In the C panel, 2,829 single-cell transcriptomes from 4 parasite blood stages were separated. The colors are the same as in B, so they were able to distinguish the different life cycle stages based on the transcription of the individual cells. These are the references that I used in preparing this presentation. For more details on aspects of single-cell sequencing that I did not have time to talk about, I recommend watching the video by Eric Chow from UC San Francisco. Thank you.